So, this is the first of two lectures where we're going to look at the molecular mechanisms that underlie the Drosophila development processes that we've already looked at. And today we're going to be looking at DNA and RNA and transcriptional hierarchies. What are our learning objectives? Well, we're going to understand how transcription factors work. And to do this, we're going to find out about the muscle transcription factor, MyoD. We're going to understand how bicoid causes gap gene expression. Can someone mute your computer? That would be really helpful. Understand how bicoid causes gap gene expression, and then understand how gap genes cause the expression of pair rule genes. So the question we want to ask is, how do we make a fibroblast into a muscle cell? And here we have a fibroblast, which is a connective tissue cell. They're extremely widespread around the body. And we want to turn these into muscle, either skeletal muscle, smooth muscle, or cardiac muscle, a very different cell type, which has one very interesting, particularly in the case of skeletal muscle and cardiac muscle, one interesting feature is that these are multiple cells, multiple polynucleate cells fused together, and this allows the formation of fibers that can contract. So what we could do is we can treat these fibroblasts with a drug called 5-azacitidine. And if we treat fibroblasts with this drug, after 2 weeks, strange things start happening in our cultures. We get the formation of different cell types, somewhat randomly. So here are our base fibroblasts, and here are some fibroblasts that have turned into fat cells or adipocytes. Here we can see the formation of muscle cells. We can see these polynucleate or multinuclear tubes that is indicative of the formation of muscle cells. And also cartilage cells or chondrocytes were also produced just by 2 weeks of treatment with 5-azacitidine. So when we look at what happens when we add 5-azacitidine, this is changing the fate of these fibroblast cells and actually driving it towards a muscle cell fate. How can we tell this is the case? Well, these are fused cells treated with 5-azacitidine, and we can see here that they are stained in one contiguous region with this marker of muscle cells, NCAM. And so we can have fusion between muscle cells, which are already here, and HeLa cells, which are a non-permissive form. We treat with 5-azacitidine, and both the cell types are able to fuse. So this shows us that our HeLa cells can turn into muscle cells in the same way that fibroblasts can also turn into muscle cells. So our induced cells that are treated with 5-azacitidine are more likely to become muscle cells. So what is 5-azacitidine? 5-azacitidine is a teratogen. So remember cyclopamine, it can cause developmental defects. Remember cyclopamine caused holoprosencephaly in sheep. So it's a teratogen, and it can also induce transformation. Remember, activated RAS, the mutant oncogene, which can make cells proliferate in an uncontrolled fashion. What does 5-azacitidine do? Well, it's a nucleoside, which means it's made of a sugar and a nitrogen containing ring, and this can bind and remove methyl groups from DNA. And why are methyl groups so important? Well, access to DNA is mediated by two forms of methylation. We have the methylation of nucleosomes, which is able to silence and condense the chromatin. Remember, when we acylate nucleosomes with a 2-carbon acyl group, if these tails become unmethylated, then we open up the chromatin. So one level is that chromatin methylation causes inability to access the DNA. But unmethylated DNA, in addition to this, is open to transcription, and unmethylated DNA can be actively transcribed. So there's at least 2 steps happening here when we treat with 5-azacitidine. Cells are made more able to have multiple fates, and this is a combination of open chromatin conformation and also the demethylation of the DNA itself, which prevents it from being silenced, and then it's another step towards it being actively transcribed. And then transcription factors induce a specific cellular fate, in this case fibroblast becoming muscle. So how was the muscle fate transcription factor MyoD discovered? Now remember that strategy of finding RAS, where we take all the DNA from a human tumour and place it in mouse fibroblasts and see where the foci are formed, and then clone the human DNA from that foci. It's a similar idea. We make libraries of cDNAs where we've treated our fibroblasts with 5-azacitidine, and we've also treated, we also have a probe for myoblast specific. So we have our probe that makes myoblasts treated with 5-azacitidine, so our fibroblast to myoblast. We also have a myoblast specific library, and then we ask the question, what are the common genes in these 2 libraries? And the common genes in these 2 libraries, we express these cDNAs in cells that are not normally permissive to make muscle cells, so a fibroblast cell line. The stain with crystal violet shows us where there are colonies of cells growing, and then we stain for myosin, which is a muscle marker, and this determines whether these cells have converted to a muscle fate, and taken together we can find colonies that have converted to a muscle fate only when we transfect, we put in MyoD into these fibroblasts. So just to recap, because this is a little bit complicated, we make myoblasts artificially by treating with 5-azacitidine. We make a myoblast specific library made from a line that specifically grows myoblasts. We probe a library that contains all the genes in our myoblast induced line, this provides us with specific clones that, when they are common, are the genes that may induce muscle fate. And in this case, we can see that MyoD induces muscle fate. In this immunostain, we can see the production of MyoD. So MyoD is both necessary and sufficient to induce muscle fate in fibroblasts. So what is MyoD? It's part of a transcription factor pathway that determines muscle fate. And so first of all, we have PAX37, which takes a non-myogenic cell and specifies it down the myogenic lineage. Then we have a combination of two transcription factors, MYF5 and MyoD, and together these are myogenic determination factors which determine further down the path of myogenic differentiation. And then finally, these two transcription factors, myogenin and MRF4, are key transcription factors that allow for this cellular fusion and making of muscle cells to occur. So there are multiple functions of transcription factors. One, they can bind DNA to make open or closed conformations of chromatin. Two, they can bind DNA to promote or inhibit the binding and function of RNA polymerase, specifically RNA polymerase II, which causes the transcription of messenger RNA. Three, they can bind DNA to enable or prevent other proteins binding to DNA to enable functions 1 and 2. So normally these things sit in massive complexes around the DNA that what needs to be transcribed. And in the human genome, there's just less than 2,600, approximately 10% of the genes in the human genome are transcription factors. So what does MyoD do specifically? Well, MyoD is in the family of helix loop helix transcription factors. It binds E-box regions in the DNA, which can be thought of as placeholders in open chromatin. So if there's open chromatin, then MyoD will bind this open chromatin. And what this allows is the recruitment of the normal basal transcription machinery, so P300, RNA polymerase II, etc., to muscle-specific promoters to allow their transcription. This is called a pioneer factor. So MyoD is a pioneer factor. It's the first transcription factor on that piece of DNA that allows subsequent transcription to occur. So let's look at two other transcription factors here, in Drosophila specifically. We have bicoid and we have caudal. And remember that bicoid had multiple effects across the anterior-posterior axis based on the concentration of bicoid. Caudal, remember, was the mRNA was at a constant concentration across the embryo, and remember, bicoid inhibits the caudal RNA and prevents its translation, and so we have caudal having a single effect of make abdomen, and bicoid having multiple effects. Now, how is it that bicoid can exhibit these multiple effects? How is it that a single transcription factor can cause multiple effects, i.e. like a morphogen? So the further away we are from the point of synthesis of the transcription factor, we get different threshold concentrations experienced by the nuclei, and these induce different downstream effects. Now, what is the mechanism of this process? So, bicoid binds the hunchback promoter. So, bicoid is an anterior-posterior polarity gene, and bicoid binds the hunchback promoter, and the hunchback gene is a gap gene. And what these guys found is that not only was it a positive regulator of hunchback transcription, but it was very particular in its function. So here are three different experiments which together show that bicoid binds the hunchback promoter. The first experiment we can perform is something called a southwestern, where you run DNA on a polyacrylamide gel, and then you incubate protein, the bicoid protein, and it binds to the hunchback gene, or to DNA that binds bicoid. You can take these DNA fragments, A, B, and C, find out by high restriction digest mapping where in the Drosophila genome these 6 bicoid binding sites are. Then we can activate, check to see whether they activate transcription by using bicoid knockout embryos and wild type embryos, and using this very old style assay called a chloramphenicol acyl transferase assay, which basically asks the question, can we get transcription of this important reporter gene, chloramphenicol acyl transferase, or CAT, from these fragments of the bicoid promoter that have been made of different lengths. So here are the different length promoter fragments attached to CAT. Here they are in bicoid mutant embryos, and we can see here very little promoter activity. And we can see here in our wild type embryos that express sufficient levels of bicoid that we see, except for the very smallest fragments which don't contain any of the presumptive bicoid binding sites, we see a large upregulation of bicoid specific CAT expression. So taken together, these 3 experiments tell us that bicoid binds specific regions of the hunchback promoter. So in this case, this specific sequence that was found multiple times is the bicoid target sequence. And this has a lot in common with other genes in Drosophila. So it has a similar common region to the Antennapedia target sequence. Remember, that's the HOX gene that misspecifies antenna as legs. And the common region here, this TAAT domain, is the homeobox domain, and this is common to a large number of transcription factors, including the majority of early transcription factors in Drosophila development. So now, how is it that different levels of bicoid can cause different effects? Well, we can determine this by doing a bicoid dosage increase experiment. And this experiment shows that bicoid controls the expression of hunchback. So we have a hunchback reporter, which tells us what the activity of hunchback transcription is, and then we can do antibody staining to determine where the hunchback protein is in space. And we can look at different promoters of different that contain different amounts of the hunchback promoter, uh, and using these different promoter fragments, we can show that if there are more binding sites, we get increased extent of the reporter because there's more bicoid binding, and because there is more bicoid binding, we get more hunchback protein being expressed. And this is proportional to the amount of copies of bicoid that are being expressed. So normally we have 2 copies of bicoid. If we have 1 copy of bicoid, we have reduced amounts of activation of the hunchback reporter. If we have no bicoid copies, then we have very reduced, almost 0 levels of hunchback reporter, and only our maternal hunchback expression is present. And then if we have very large numbers of bicoid copies, if we have 6 copies of the bicoid protein, then we get much more extensive expression, activation of the hunchback reporter, and much more extensive expression of the hunchback protein. And this demonstrated quite nicely that bicoid acts as a what they call gradient morphogen and is a concentration dependent transcriptional activator. Now, how is it that we can still form these gradients of hunchback expression? An experiment that shows this is looking at the whole of the promoter for hunchback, and we can see here it has 2 different kinds of binding sites. A1, 2, and 3 are high affinity binding sites which can bind in low concentrations of bicoid, and X1, 2, and 3 are the low affinity binding sites which can only be bound in high concentrations of bicoid. And we can see here that with our normal hunchback promoter, which has both the high and low affinity binding sites, we see normal high levels of expression of hunchback and the gradient coming off anterior to posterior. If we look at just a fragment that contains high affinity binding sites, we get lower levels of expression overall, but they're spread to the right correct region of the embryo. And X1, 2, and 3, the low affinity binding sites, which bind bicoid less efficiently, if we only have this, then our LAC-Z reporter gene is only expressed in the very anterior regions. This is telling us that different binding sites, different bicoid binding sites of different affinities, different capabilities of binding, define the hunchback expression domains. So again, here is our normal promoter. We get our normal bicoid pattern of expression. And we have a high affinity promoter, which binds bicoid better than the low affinity promoter, which binds bicoid less well. So bicoid binds better to the high affinity promoter than the low affinity promoter. Or in the low affinity promoter, you need more bicoid to cause the same effect, which is hunchback transcription. So the key points here are more bicoid, more hunchback expression. And different binding sites have different binding efficiencies. So here with our normal wild type expression of bicoid, we get our normal expression of hunchback. If we have no bicoid, we have no hunchback expression. Now, if there's no gradient, it doesn't matter if we have increased dosage of bicoid. If there's no gradient to this bicoid, then we still see contiguous expression across the whole embryo. So we need that gradient of bicoid to define the clear region where hunchback is expressed in the embryo. So remember I was talking about a Drosophila transcription hierarchy, and this is how networks of transcription factors interact with each other to form the expression of downstream transcription factors. And remember that transcription factors can repress as well as activate expression. So here we have our maternal RNAs, bicoid, hunchback, caudal, and torso, and then these give rise to the maternal proteins, have bicoid transcription factor, hunchback transcription factor, caudal, and torso. And we can see here that bicoid activates the transcription factor of the gap genes of crippled and knurped, but we also find that hunchback can inhibit the expression of these gap genes. And so it is that there's a network of mutual activation and inhibition, and this can tightly define the different regions of gap gene expression in the Drosophila embryo. So hunchback represses the expression of crapple and knurps, which are two of these important anterior transcription factors. So when we have normal hunchback expression, here in our wild type, we see normal expression of crippled and knurps in discrete domains. If we don't have any zygotic, so this is a hunchback zygotic mutant, so we get maternal hunchback but not hunchback transcribed by the embryo after the mid-blassular transition. When we have no zygotic hunchback expression, the cripple and knurp zones expand and overlap. And we can see this here by this crippled in situ hybridization. We can see an increase in the domain of expression of crippled. And here in the knurps in situ hybridization, this second domain expands and begins to overlap with the other domain of knurps expression. So hunchback represses both cripple and knurps expression. So this is a good time to think about what's happening in this hierarchy of transcription. And remember that until the segment polarity genes are expressed, we're a cellular blastoderm. So in mid-blassular transition, we begin to get transcription of these gap domain genes controlled by the maternal gradients that are set up in the developing oocyte and then translated in the developing embryo. And then what's very interesting is that each pair-rule gene domain is differentially controlled by a combination of gap genes. And what you can see here is that these pair-rule domains overlap different numbers and different located gap domains. So each pair-rule domain is differentially controlled by a different combination of gap genes. And then that gap gene expression sets up, our pair-rule gene expression rather, sets up the arrangement of parasegment boundaries where cells are talking to each other to determine identity marking the parasegments, which are the developmental repeating unit. So here we have our pairal gene expression, and this is even-skipped and Fushi-Terazu, or even-skipped and odd-skipped, and we can see this alternating pattern of expression of these transcription factors, accurately spaced. They have sharp anterior boundaries that delineate the boundaries of the parasegments. So how, for instance, is Fushi-Terazu expression controlled specifically in this segment, in this region? So this experiment is all about the control of one of these stripes. We're going to call it even-skipped stripe 2. And to get an idea of what might be controlling even-skipped, it's a really good idea to look at the previous hierarchies of transcription factors, so our gap genes and our maternal genes, and where they might be expressed. So this arrow is pointing to the stripe 2. And we can see here by this antibody co-immunofluorescence stain that even-skipped and hunchback co-localized. We can see the yellow band here. Now, Krüppel is expressed to posterior, so it doesn't overlap with even-skipped expression. Neither does giant, so we have this band here that specifically overlaps a region of high bicoid and high hunchback, but there's no Krüppel and no giant. So understanding the expression pattern over this stripe can help us understand the control of expression of this specific even-skipped stripe. And so we can identify the specific region of the even-skipped promoter that controls stripe 2. And we can find this bit here in this stripe 2-7 region, and then we can make a LAC-Z reporter gene that we can put into Drosophila. And then we can use different mutants. We can use a Krüppel mutant or a giant mutant. And remember, regions that have no Krüppel and no giant, but still have high levels of hunchback or bicoid, are the regions where this even-skip stripe will be expressed. So in our normal wild-type fly, we have this very discrete stripe expressed, and this is our LAC-Z reporter. If we have no Krüppel, we have an expanded domain posteriorly, that's increasing the size of the stripe. And if we have no giant expression, then obviously we will have a stripe whose expression is expanded anteriorly. So Krüppel and giant inhibit the expression of the stripe 2 even-skipped. Loss of Krüppel or giant extends stripe 2 where the transcription factor is lost, and this is indicated by this blue arrow. No Krüppel, so Krüppel mutant, we get expansion posteriorly, and no giant, with our giant mutant, we get expansion anteriorly. So these activating and repressing transcription factors overlap. And back in 1991, we would use a very old technique called Maxine-Gilbert sequencing and pair this with a DNase protection assay. We add a protein, either bicoid or Krüppeled, to radiolabeled DNA, and then partially digest this DNA with a DNase. This DNase 1. Then we can run this on a polyacrylamide gel, and the protected regions that have reduced or absent sequence, so these regions here and these regions here, are the binding sites for these transcription factors. And so you can see here that bicoid binds this region here, and crippled binds this region here, and you can see that these two regions overlap. And you can see this here. We can see that in our proximal and distal clusters, our hunchback and bicoid transcription factors overlap, binding sites overlap our giant and crippled, so our repressive transcription factor binding sites, both proximally and distally. So this concept of inhibition, activating and repressing transcription factor binding sites overlapping allows this tight control of things like the pairal gene expression in a specific stripe. So that is the end of the first lecture. And what did we learn? We understood how transcription factors work, either by opening chromatin, by binding to DNA to allow other transcription factors to transcribe DNA, or to transcribe DNA itself once those pioneer factors have bound the DNA. We found out about the experiments with 5-azacitidine that were able to isolate the transcription factor MyoD, which is an important pioneer factor which binds regions of DNA which allow the transcription of genes that drive a cell towards a muscle fate. We understand how different affinity bicoid binding sites, low and high affinity, cause the expression of hunchback in different regions of the Drosophila embryo. And then finally we saw how gap genes cause the expression of specific stripes of pairal genes by combinations of activating transcription by bicoid and hunchback, and inhibition of transcription by downstream gap genes such as cripple and giant, and how this is caused by these activating and repressing transcription factors competing for binding in the same region of the DNA. So the next part of the mechanisms of development is the concept of parasegment identification. And as we can see here, here is our early development expression of pairal genes, here Evenskipped and Fitsutaratsu. And then we can see here that they get tighter and tighter domains of expression. Now this tightness of domain expression, where we have clearly defined parasegments, and hence our segments are clearly defined, requires cell-cell signaling. And that's what we're going to be looking at in the next part, the next lecture after our short break. So that's the end of this lecture. We'll start back at 5 before the hour. Okay?